Every single cell in your body is running a chemical cycle right now, like at this exact second. It's churning away, producing the energy that's literally keeping you alive. It's called the Krebs Cycle. Now, let's be real. Most students absolutely hate it. Not because it's actually that hard, but honestly, because nobody ever explains it simply. Well, until now. So let's dive right into this. The Krebs Cycle, which, by the way, you might also hear called the Citric Acid Cycle or the TCA Cycle, is basically the beating heart of your cellular metabolism. It's just running continuously in the background of your life. Every breath you take, every move you make, it all relies on what we're about to cover. Here's our game plan for today's explainer. First, what is it? Second, location and setup. Third, inputs and outputs. Fourth, we'll walk through the 8 steps. And finally, an exam survival guide to lock it all in. All right, part 1. What actually is this thing? So your body needs ATP, right? That's the ultimate energy currency of your cells. If a cell wants to do any work at all, it has to pay up in ATP. Now, the Krebs cycle itself doesn't actually make a ton of ATP directly, but, and this is absolutely crucial, it powers everything that does. Think of it like the engine room of the cell. To really get why it's so important, let's quickly compare it to the step right before it, glycolysis. Glycolysis is anaerobic, meaning no oxygen is needed. It happens out in the cell's cytoplasm, and it kind of just partially breaks down a glucose molecule into 2 molecules of pyruvate. But the Krebs cycle, it's aerobic. It needs oxygen to run. It takes place deep inside the mitochondria, and its whole job is to complete the breakdown of that pyruvate, extracting every last bit of potential energy. Looking at our overall roadmap here, cellular respiration is really a 3-step journey. Glycolysis is step 1. The Krebs cycle, which is step 2, is the crucial bridge. It's prepping all these high-energy electrons that are going to be absolutely vital for step 3, which is oxidative phosphorylation. Moving on to section 2, location and setup. You know, in biology, location is everything. If you don't know where a reaction is happening, you're missing half the story. Take a look at the anatomy of the mitochondria here. See that fluid-filled center? That is the mitochondrial matrix. The Krebs cycle happens entirely in the matrix. Not the cytoplasm, not the intermembrane space, the matrix. Seriously, if you're taking an exam on this anytime soon, write that down right now. It's practically a guaranteed question. The matrix has all the water, the enzymes, the coenzymes, basically everything this cycle needs to keep spinning. Okay, section 3, inputs and outputs. Let's look at the biological blueprint here. What goes into this engine room and what exactly comes out? So before the cycle even starts, the pyruvate from glycolysis gets converted into a 2-carbon molecule called acetyl-CoA. This acetyl-CoA, that's our input. It's basically the ticket to get on the ride. Once 1 molecule of acetyl-CoA enters, the cycle completes 1 full turn. And for every single turn, it spits out 4 really critical things. First, it releases 2 molecules of carbon dioxide, which, fun fact, is literally the CO2 you are exhaling right this second. It also produces 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of ATP, or GTP, depending on the specific cell. Those NADH and FADH2 molecules, they're electron carriers. Think of them like tiny biological batteries just glowing with captured energy. But wait, the absolute most crucial thing to remember here is multiply by 2. Remember how 1 molecule of glucose was split into 2 molecules of pyruvate during glycolysis? Well, that means you get 2 molecules of acetyl-CoA entering the cycle. So for every single molecule of glucose you eat, the Krebs cycle has to turn twice. That means you've got to double every single output we just talked about. 1 glucose yields 4 CO2, 6 NADH, 2 FADH2, and 2 ATP from this cycle. Whatever you do, never forget to multiply by 2. All right, section 4, the 8 steps. Let's get into it. This is where the actual chemistry happens. And I know, at first glance, it looks like a terrifying alphabet soup of complex words. But don't worry, we're gonna break it down so it makes perfect sense. First up, I have to share this hilarious, but honestly highly effective memory trick I found straight from a biology student Reddit thread. Officer, can I keep selling sex for money? Yeah, I know, it's totally ridiculous, but it lines up perfectly with the first letters of the metabolites in the cycle. Oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, and malate. Just write that phrase at the top of your scratch paper during a test, and I promise you, you will never blank on the order of these molecules again. So let's look at the cycle itself. What I really want you to focus on here are the numbers of carbons, the C's, over on the right side. Notice how we start with 6 carbons, then drop down to 5, and finally down to 4. The whole first half of this cycle is really just about tearing carbons off the molecule to release energy. Let's walk through exactly how that plays out. Step 1. Our 2-carbon input, acetyl-CoA, enters and joins forces with a 4-carbon molecule called oxaloacetate. 2 plus 4 equals 6, right? So this forms a brand new 6-carbon molecule called citrate. And hey, that's exactly why it's also called the citric acid cycle. Moving to step 2. Citrate gets slightly rearranged into an isomer called isocitrate. Then step 3, this is where the action really starts. Isocitrate gets oxidized. It loses a carbon, which just floats away as CO2, and it transfers high-energy electrons to our first carrier, creating our very first NADH molecule. So now we're left with a 5-carbon molecule called alpha-ketoglutarate. Step 4, we basically do it all over again. Alpha-ketoglutarate loses another carbon as CO2, creates our second NADH, and a coenzyme A gets attached. This leaves us with a 4-carbon molecule called succinyl-CoA. So, just like that, we've stripped away 2 carbons and captured some serious electrical energy. Okay, now for the second half of the cycle, where we stay at 4 carbons and really just focus on resetting the whole system. Step 5. The coenzyme A on succinyl-CoA gets ripped off, and the energy released from that is used to directly produce 1 molecule of ATP, or GTP. Now listen closely. This is the only step in the entire cycle that makes ATP directly. Got it? Okay, so now we're left with succinate. Step 6. Succinate gets oxidized. It transfers hydrogen atoms to another carrier, turning FAD into FADH2 and forming fumarate. Step 7. A water molecule gets added to fumarate to form malate. And finally, step 8. Malate is oxidized to form oxaloacetate, creating our third and final NADH molecule in the process. And would you look at that? We have completely regenerated oxaloacetate, which is the exact 4-carbon molecule we need to combine with the very next acetyl-CoA that comes along. The cycle is completely reset and ready to spin all over again.
Moving on to Section 5, the Exam Survival Guide.
All right, take a deep breath. I know that was a lot of chemistry, but if you're studying for a test, here are the 5 things you need to know absolutely cold. Number 1, location. The Krebs cycle happens in the mitochondrial matrix. Number 2, the outputs per turn. Remember, 1 turn yields 2 CO2, 3 NADH, 1 FADH2, and 1 ATP. Number 3, the multiplier. The cycle runs twice for every 1 molecule of glucose, so always double those outputs for your total yield. Number 4, know the key players by name. The cycle begins when acetyl-CoA combines with oxaloacetate to form citrate. And finally, number 5, the ultimate destination. All those NADH and FADH2 molecules you just made, they actually aren't used here. They're packed full of high-energy electrons and headed straight for the electron transport chain. Which brings us to the end of our explainer today. Let's do a really quick recap. The Krebs cycle is step 2 of cellular respiration. It occurs in the mitochondrial matrix, and it turns twice per glucose. Simple enough, right? But it does leave us with a massive cliffhanger. Where do all those glowing blue NADH and purple FADH2 molecules actually go next? How do they eventually turn into the massive payout of ATP that your body desperately needs to survive? Well, they go to oxidative phosphorylation, the final step. And we are going to cover exactly how those electron carriers finally become ATP in our next explainer. Make sure you subscribe so you don't miss it.